Uh, Thank you for to my second who did that for me. So hello and welcome everyone to our second trainee research presentation series of 2021. Always a great way to give trainees of the Women's Health Research Cluster a stage to share their research with us, a great way to learn about exciting things that other trainees in the cluster are doing, a great way to form collaborative opportunities, and also a great way just to practice talks and put that on your CV. So all of you better put that on your CV. I will talk later on about our upcoming trainee research presentations that are coming out throughout the year. But one of the first things I would like to do is to acknowledge that we live, work, and play and participate in community and on unceded and ceded and traditional territories of the 203 First Nations, along with 38 Métis chartered communities, each of which possess their own unique traditions and history here on this land that we now refer to as, where I am, British Columbia. We acknowledge the importance and Truth and Reconciliation Commission of Canada's Call to Action and the United Nations Declaration on the Rights of Indigenous Peoples and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. And today, as I said before, I'm joining you from Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. And in case you were wondering where you might be able to donate to any of these causes for Indigenous peoples and Indigenous women's health, I'll be sharing a link in the chat near the end of our presentations while making other announcements. So thank you for that. I'd also like to acknowledge our sponsors. Of course, this event wouldn't be possible without contributions from our sponsors. And also, if you like what you hear from these presentations, feel free to tweet about it and tag us in those tweets and with the hashtag TraineeBlitz throughout the presentation. So yes, so we can get started. I'm not going to be rushing anyone today because we are down one presenter, but we will start nicely with Stephanie, Dr. Stephanie Koebele. Stephanie holds a PhD in behavioral neuroscience in psychology from Arizona State University. Her doctoral research broadly investigated reproductive hormones, menopause, memory, and aging using rodent models. Her dissertation focused on understanding the impact of hysterectomy on the trajectory of cognitive aging. And Stephanie is currently a postdoctoral research scholar in the Center for Evolution and Medicine at Arizona State University, where she will investigate environmental endocrine immune interactions in the context of human chronic diseases of aging, including Alzheimer's disease and related dementias and cardiovascular disease from an evolutionary perspective. And today, Stephanie will be presenting a talk titled Hysterectomy Uniquely Impacts Spatial Memory in a Rat Model, a role for the non-pregnant uterus in cognitive processes. So please take away, Stephanie. I'm very excited to hear your research.
Thanks. All right. All right, so thank you for joining today and to the Women's Health Research Cluster for having this event and allowing me to share my research. I'm going to jump right in to talk to you about my dissertation research on hysterectomy and cognition, but I just want to acknowledge that all of the research that I am discussing today was completed as part of my doctoral studies with Dr. Heather Bamonte-Nelson at the ASU Department of Psychology, and this work is actually published in the journal Endocrinology. So while most women undergo a natural transition to menopause, there is a significant number of women who undergo gynecological surgery, which can happen at any point in their lives, and that can lead to surgical menopause. This graph on the right indicates that bilateral oophorectomy before natural menopause can lead to up to a 70% increased relative risk of developing cognitive impairment or dementia And what really struck me when I looked at this graph is that hysterectomy alone with ovarian conservation before the age of 50 also appears to increase this relative risk of developing dementia. So given that hysterectomy is the second most common gynecological surgery, second only to cesarean section, this really piqued my interest and drove my dissertation research, which essentially boiled down to a question: what about the uterus? In many clinical menopause studies, women with ovaries are often grouped together, whether or not they have a uterus. And when cognition is considered in the context of surgical menopause, ovarian changes are generally considered to be the primary cause for any altered cognitive functioning that occurs. And this is largely due to the dogma in the field that without hormone stimulation, the uterus is a quiescent and to a large degree useless organ, and that the sole function of the uterus is to accommodate and support a fetus. So I probably don't have to convince this audience that that likely is not the case. But based on the research from Mayo Clinic that I showed you on the last slide, not moving forward, I hypothesized that hysterectomy with ovarian conservation could differentially impact cognition compared to a fully intact reproductive system. So I developed a novel surgery protocol in our lab along with the vets at Arizona State, where I basically had 4 surgical groups: a sham group where they underwent surgery, anesthetization, etc., but their reproductive tract remained intact; an ovaryectomy group where I just removed the ovaries; a hysterectomy group where I just removed the uterus; and then an ovex plus hysterectomy group where I removed both the uterus and the ovaries. The rats had the surgery at 5 months of age, so they were considered adults at the time of surgery. And then 6 weeks after the surgery, we tested them on a battery of spatial tasks that assessed working memory and reference memory. I don't have time to get into the details of that, but if you're interested in those tasks, you can check out the paper. I'm going to be sharing a interesting finding from the water radial arm maze. So on the y-axis here, we have errors, and on the x-axis, you can see the surgery groups. And during the asymptotic phase of testing, which is at the end of testing after the animals have learned the rules of the task, we found a main effect of surgery where hysterectomy made more working memory incorrect errors than all other treatment groups. And when we looked at their performance across trials, we found a marginal surgery by trial interaction. So the water radial arm maze is the type of task that gets more difficult as trials progress. And so we planned to look at what we call the high working memory load trials. And again, we found a main effect of surgery where hysterectomy animals made significantly more errors and therefore had poorer performance compared to all other groups when working memory load was highly taxed. So this was the first systematic investigation into variations in surgical menopause, including hysterectomy in a rat model. And hysterectomy with ovarian conservation in adulthood negatively impacted spatial working memory compared to sham, ovex, and ovex plus hysterectomy rats 2 months after surgery. So collectively, these findings were the first to demonstrate or suggest that the non-pregnant uterus is not a dormant organ and may in fact play a key role in cognition. So after our findings were published in the journal Endocrinology, it was picked up by the media, which was very exciting to see the conversation that was sparked online, and particularly that so many people were sharing their stories and feeling validation about their experiences following hysterectomy, and how important this preclinical research is to our understanding of women's health as a whole. And so I actually received a predoctoral NRSA that evaluated whether these cognitive changes we saw in this study were transient or long lasting. I can't share that with you quite yet, but we are getting ready to submit that for peer review, which I'm very excited about. And a future direction for the lab is also to investigate the neurobiological mechanisms underlying these cognitive behavioral effects that we observed. So as Travis mentioned, I have since graduated and switched to a new postdoc in the laboratory of Dr. Ben Trumbull at ASU still, but in the Center for Evolution and Medicine. And broadly, that lab investigates environmental, endocrine, immune interactions as risk factors for chronic diseases of aging from an evolutionary perspective through the Chumane Health and Life History Project. And my particular focus in this lab will be on reproductive hormone fluctuations across the lifespan, as well as impacts of pregnancy, chronic immune challenges, and menopause on cognition in a new species to me, humans. So with that, I just want to say thanks again to the Women's Health Research Cluster for having this event, to Dr. Heather Monty-Nelson for supporting me during my doctoral studies, and to all of you for being here today.
Fantastic. Thank you so much, Stephanie. Everyone who wants to give a digital clap or a digital party streamer, feel free over this digital platform. Amazing. Thank you so much, Stephanie, for your talk. I forgot to mention before the talks began that for questions and things, feel free to put questions into the chat and I can just read them out. Or since we are such a nice tight-knit group, feel free to raise your hand and I can unmute you if you have a question as well. And while people are thinking of questions and while questions are coming in, I, of course, have many a question for you. One of which I'm just wondering, so of course, 5-minute presentations, you can't get into too much of the nitty gritty. I'm very interested in, because you mentioned neurological mechanisms, and I'm interested in what neurological mechanisms are you thinking may play a role, and what role maybe that hormones might play, especially when looking at OVX versus hysterectomy?
Yeah, so that is a great question, and just something I couldn't address in 5 minutes, but part of my NRSA was actually starting to look into potential neurobiological mechanisms. My original idea was that there's some sort of dysregulation between the hypothalamic pituitary reproductive tract axis when the uterus is removed that is not secondary to the ovarian connections. So I was originally thinking maybe the gonadotropins were altered. I wasn't able to find an assay that I could test that during my PhD, but another idea that I have is also that there are direct autonomic connections between the uterus and the brain, because as we know, the brain has a big role in parturition and across the menstrual cycle. And so I think maybe that the disruption of those autonomic connections might be further impacting, neurotransmitters, maybe dopamine, other acetylcholine, so I have lots of ideas, but, this is something I'm hoping the lab will carry on because it's really exciting stuff.
Yes. And to add on to that too, because you had tissue collection that occurred as well, correct? Well, I guess, and did you take blood samples, plasma samples as well to maybe look at things like
yes, so I, again, just didn't have time to include that, but one really cool feature of this paper was that we actually took, we got serum hormone levels and we got ovarian follicle counts. And also we took vaginal smears. And so the vaginal smears showed normal cycling in the hysterectomy animals compared to the sham animals. So they seemed normal at least 6 weeks after surgery. And then the actual ovarian morphology was not different between the sham intact animals and the hysterectomy animals. So to me, that really indicates that that it is something that's happening with the uterus and not the ovaries. We also did take blood, and in this published study, we reported some marginal differences in steroid hormones, as well as a difference in follicle stimulating hormone, but I actually was not able to replicate that change with the follow-up study. So I have some hesitations about whether, those serum effects are real or not, but you will see in the publication, we do report some hormone changes there.
Awesome, thank you so much. Awesome research. All right. Well, we should move to, I don't see any questions, so let's move to our second presenter of the day, and we can get to even more questions even later on after we're done all of these presentations. So next up, we have Eunice Bawafaa, a healthcare professional who has worked in a materially under-resourced country in the global south for several years. Specifically, she has worked as a counselor for pregnant women living with HIV/AIDS and their partners, which has piqued her interest in issues of nurse-patient empowerment in relation to achieving quality nursing care. Eunice is passionate about nursing care that seeks to sustain social justice activism in health education and healthcare provision. She is interested in advancing the need for more emphasis on restructuring the nursing education system to include respectful care for marginalized women and to enhance research related to women's health. So today, Eunice will be presenting a talk called Women's Experiences and Perception of Reproductive Health Access in Rural Ghana. So yes, take it away, Eunice, and I will be presenting Eunice's presentation for her. So just a second.
Okay, thank you so much, Travis, for that wonderful introduction. I must mention that I'm joining you from Ghana, and my apologies for not being able to join with my video. I have internet issues and I really want to keep it stable. So my apologies for that. Next slide, please. In contextualizing this study, Ghana is a former colonized study, and from the outset, colonialism displaced and erased existing institutions in the colonies and imposed European institutions. And for women particularly, their oppression was multifaceted in the sense that not only were matriarchal societies transformed into patriarchal ones, but women's bodies and sexualities were assaulted systematically. And women also lost autonomy, not only over productive and economic resources, but perhaps more detrimentally over their bodies and reproductive matters. And particularly in Ghana, women's identities generally revolve around childbearing, motherhood, and reproductive labor. And sadly, women, including myself, have constructed our own identities along these lines. And as to how all this impacts women's health, in the period following colonialism in the third world, in the health sector and to date, expertise about women's bodies and health, especially during and after pregnancy, have been yielded to medical personnel. And there's the assumption that the health personnel know best and need not consult the patient. And this is a major form of marginalization that actually impacts women's dignities and psychological well-being. And in reality, there's a total disregard of our spiritual midwifery healing practices and wisdom of the elders, which have been handed down over generations, and this has strong impact on the outcome of reproductive health. So consequently, the intersecting issues of poverty, gender, religion, and patriarchal norms contribute to subjugating women in ways that prevent them from accessing reproductive health care. Next slide, please. So what is the problem? In Ghana, women's knowledge and experiences with reproductive and maternity care are discounted in the face of Western biomedical practices, values, and knowledge. So the preference of Western practices has led to the alienation of local knowledges and traditions. And this has led to women in the global South experiencing poor treatment in the healthcare system. And in particular, this has created a problem of poor access of maternal mortalities. So due to the trust that is imposed on nurse leaders and nurses by these communities, nurses can potentially play a significant role in improving reproductive health services, but there's limited research evidence available in this regard to guide nurses in our practice and service delivery. Therefore, in the nursing profession, the problem of poor access is not well understood, and in particular, we do not know much about the experiences and perceptions of the rural women themselves when it comes to reproductive health access. Next slide. So the purpose of this study is to show how a contextualized explanation of women's experiences and perspectives of reproductive health can provide direction for nurses' future efforts in improving reproductive health access. Next slide, please. So for our significance, we are hoping that findings can form a theoretical basis for supporting and understanding reproductive health access, and it will also shed light on the barriers and facilitators that contribute to reproductive health and also improve reproductive health outcomes. Next slide. So what are my research questions? So I am asking, what are women's experiences and perceptions of reproductive health care in rural Ghana? What is the role of gender, power relations, and sociocultural factors in shaping women's perceptions and experiences of reproductive health care? And what are the key priorities for nurses to consider in their efforts to improve reproductive health access? Next slide. For the methodology, the underpinning theory is the critical social theory, and I chose this theory because it's primarily concerned with logical opinions and discussions around experiences that are socially constructed, and it also focuses on the manifestation of power relations that are produced by social and historical discourses. And for the design specifically, it's a focused ethnographic study that will make use of interviews and focus group discussions. Ethical approval, of course, will be sought from UPC Ethics Board and the Research Ethics Board from Adabraka, Ghana. Study participants and community advisory group will be compensated for their time at the end of the study, and data certainly will be collected and analyzed using qualitative data methods. Next slide. For the possible limitations, there's the eligibility for bias on met timelines, data losses, and maybe participants dropping out of the study. Next slide. For knowledge integration, there's patient engagement involving rural women, the community advisory group, and other stakeholders. And the possible beneficiaries to a large extent are the community members who will have improved reproductive health services. And we're also hoping that you'll have autonomy and ownership over the study and its findings. And findings from this study will be made available to various stakeholders in an attempt to encourage dialogue on collaborative and creative community action for change and transformation, especially in reproductive health access. Next slide. So I conclude by saying that reproductive health is nested in a web of intersectional factors, and to holistically understand women's experiences and perceptions of reproductive health care, you need to examine all other factors so that nurses can actually move beyond the focus of the individual to promote a wider understanding of multiple perspectives on competing factors influencing wellness and well-being of women in reproductive health access. Next slide. Thank you, and I'm hoping to be back to share the results of this study with this group when it is done. This is only a proposal. Thank you.
Awesome, thank you so much. Let me close it. Perfect. Yeah, let me put out my party streamers here. Awesome, thank you so much, Eunice. Does anyone have any questions for Eunice before I jump right in? All right, well, I do have maybe a couple questions. One question that I have for you, Eunice. I'm wondering, so you are looking at the individual, and I'm wondering if you have plans for examining a certain age group and an idea of why examining a certain age group might be better for, well, not even better, but might get you specific data that you require for your collection.
Yes, I'm actually looking at so we are going to have the individual interviews and then a focus group with them, but at the same time, we are going to have a community advisory group that will be more like a steering committee that will oversee through the study, actually see to it that it stays on focus and on track, but data will not be collected from the steering committee, only from the study participants themselves.
Okay, perfect. And do you know what ages that you'll be collecting from?
Yes. So we are looking at 16 to the ages of 45, and that is also because 16 has been considered a normal age for marriage in rural Ghana. So that was why we took that. And we're actually looking at people who are actively in their reproductive years and will be seeking reproductive health care from the health facility or other traditional birth attendants.
Perfect, perfect. Awesome, thank you so much. Does anyone else have any questions? All right, well, thank you, Eunice. If you have any questions that come up, feel free to write them in the chat. We can get to them at the end of the presentations as well. So up next, I would like to introduce Dr. Alesia Prakapenka. Alesia is a postdoctoral researcher in the laboratory of Dr. Donna Corol at Syracuse University. Alesia completed her PhD in interdisciplinary neuroscience at Arizona State University under co-mentorship of Drs. Heather Bimonte Nelson and Rachel Siriani. During her doctoral work, Alesia developed translationally relevant delivery strategies for hormones to maximize their beneficial cognitive effects while minimizing undesired peripheral burden in a rat model of menopause. Awesome. Her ongoing work investigates the underlying biological mechanisms that contribute to hormone regulation of cognitive function in female rats across age and multiple memory systems. So today, Alesia will present a talk titled The Curves, Turns, and Bumps Along the Path of Cognitive Aging for Females. So take it away, Alesia.
Awesome. Well, thank you so much for having me here today. And I'm really excited to share some of the recent data I have coming in as a postdoc in Dr. Donna Corral's lab. So let me go on to my next slide. So primarily over the last several years, my research has focused on evaluating how ovarian hormones modulate learning and memory. So if we look at the female and what happens to her across the lifespan, we see that she will be met with a variety of different events, both endogenous and exogenous, that could alter her circulating ovarian hormone profile. And so what my goal is, I use the female rat to try to understand how these different changes in the circulating ovarian hormone will then impact her cognitive health across the lifespan. Particularly within my postdoctoral research, one additional aspect that I've tacked on top of it is looking at genetic variations. So an individual's genetic variation could actually impact their cognitive sensitivity to ovarian hormones. And so that's one of the aspects that I'm aiming to evaluate. I'm kind of zeroing in onto the topic of my data blitz here. The hormone of interest I'll be covering is estradiol, which is one of the primary estrogens found within the body for women and the female rat. And it is known to both impair and improve learning and memory, depending on a variety of factors. These include age, as well as different memory systems, things like that. Additionally, I'll be focusing on the brain-derived neurotrophic factor, BDNF for short. So BDNF is a neurotrophin that's actually found throughout the body, but specific to learning and memory, it plays a role within neuroplasticity. It seems to be beneficial for cognitive function. And several studies and labs suggest that estradiol and BDNF potentially interact together to hold their cognitive effects. And so that's one of the things that I'm trying to figure out in my work. And I do that by using a model of a genetic variation, particularly within the BDNF gene. So it's called the Val66Met single nucleotide polymorphism. And so studies suggest that this particular polymorphism decreases activity-dependent BDNF release. And so interestingly, other work suggests that estradiol increases activity-dependent BDNF release. So when you put the two together, our working hypothesis is that if estradiol regulates learning and memory through this activity-dependent BDNF release, we think that the MET carriers, the ones that express the single nucleotide polymorphism, will be insensitive to estradiol's regulation of learning and memory, since you won't have this activity-dependent BDNF release. So luckily, we actually do have a model of this polymorphism within the rat. We have collaborators at Michigan State University that developed it using CRISPR-Cas9. And so the first thing that we did is we actually just staged the reproductive cycles. We were hoping to prove that they're similar, but what we found is, so here's the estrous cycle of our young adult rats, which typically lasts 4 to 5 days. And that's what we see in our control rats, our Val-Vals. So these MET rats are the ones that exhibit this polymorphism, and their cycles are about an average a day and a half longer, which is a significant amount of time if you consider the average length of the rat cycle. When we look deeper into it, we see that the MET rats spend less time in the high ovarian hormone stages within a cycle compared to the Val-Vals, and more time of their cycle within these low ovarian hormone cycle stages compared to the Val-Vals. We further then looked at the middle age to evaluate whether this has any kind of relation to onset of reproductive senescence. And what we found is that our MET rats actually enter reproductive senescence earlier, about what we estimate to be about 3 months compared to our Val-Vals. So here we have our 11 and 12-month-old rats. And the Val-Val rats, about 70% of them are still cycling very irregularly, versus at this same age point, our MET rats are no longer regularly cycling, which is indicative to us of an earlier onset of reproductive senescence. So overall, this suggests that there's this lifetime reduction in ovarian hormone exposure that our met rats are experiencing, which could potentially lead to decreased neuroprotection from the hormones, as well as altered behavioral and cognitive profiles. So we did do some behavioral analyses with these rats as well. So for instance, we tested them on the open field where we looked at different behaviors where they can just explore their environment. And so we found that the locomotor behavior within our met rats is actually insensitive to the fluctuations in ovarian hormone levels across the estrous cycles, versus we did not see this in vals. They had different locomotor behavior depending on the stage of the cycle. We also found elevated anxiety-like behaviors within our met rats, as well as increased stress-related behaviors, as well as stress hormone levels within the met rats. So their overall kind of this behavioral profile is very different. We followed this up by looking at their adrenal weights, which releases stress hormones, right? And so we see both in young adult and middle age that our met rats have significantly heavier uterine hormone weights compared to our val rats. And this difference is actually exacerbated with age. It's about a twofold difference in young adult, and it's threefold within our middle age, which is a very drastic difference. And we're interested in pursuing this further to understand what's actually happening with this different signaling cascades that are going on with this polymorphism. And finally, we did test our rats on the dual novel object recognition to look at learning and memory. And we found an interaction between genotype and estradiol on object recognition memory. So here I have this interaction depicted in the graph. Our val and our met rats were overrectomized, and then they received their respective treatments. And here we have the recognition score. So the higher the number, the better the memory. And we see that for the val rats, estrogen tended to improve recognition memory, versus for our met rats, instead of being insensitive to estradiol treatment, it actually tended to impair their recognition memory, which is an exciting result that we didn't really expect to find. It didn't support our hypothesis, but it could be potentially this interaction between the stress hormones, the HPG axis, the HPA axis, all these differences, physiological differences that we unexpectedly found might be actually contributing to these memory effects. So what are the implications for this? Women across their lifespan do have some increased risk factors for developing certain neural diseases, and Alzheimer's disease being one of them. And a lot of these factors that I just showed you are also risk factors for developing Alzheimer's disease, such as chronic stress exposure, earlier onset of reproductive senescence. So we're trying to identify these factors, and hopefully that'll lead to strategies that can optimize women's health across their lifespan. And so with that, I would like to thank Dr. Donna Coro's lab, as well as Dr. Paul Gould's lab, which is right next door to us. Here in purple is basically an army of students that have helped me throughout working with these animals. Our collaborators at Michigan State University who provided us with the rats, and of course our funding sources and our staff that take care of our animals.
Amazing, thank you so much. Voila. Awesome. So do we have any questions right off the bat? Perfect. Well, I, of course, do. Many questions, always looking for a new thing. I love your research. Very interested in, so one question I have, are you interested in, or have you looked at neurogenesis at all? And in keeping
have
with...
Not at all. We've actually been more on the neurodegenerative side of it. No. Being honest here, we have not even considered it.
Yeah. Because I was wondering, because since you're targeting BDNF, and also since you're targeting estradiol, and both of those have both play factors into neurogenesis, would be interesting to see how that might be, I don't know, being reduced or being increased or just changed in these met animals.
Super novel work that we are still trying to figure it out. I'm hoping to get it out as a paper soon, but unpublished, presenting it at Society for Behavioral Neurotechnology next week, but really exciting.
Yes, very, very exciting. Another question I have is just which, with that replacement study, which type of estrogens were you replacing with?
It was
And
17 beta estradiol benzoate, and it was 48 and 24 hours before the actual task. And usually we wait 3 weeks between overectomy and doing the treatment. So the 48 and the 24 hours kind of mimics that increase that you would see across the cycle when during the paroestrus where it's going to ovulation. So kind of mimic that peak, and that's when we test at that peak.
perfect. And did you ever want to or think of doing any other like estrone?
Oh boy, do I.
Yes, of course.
That's kind of what I want to take with me when I hopefully eventually start my own lab, is I want to tack on the other hormones and look at the interaction. So my graduate work, some of it looked at estrogens and progestins together. So I want to kind of build on that, but now with these different, the aging profile, the different genetic profiles, it's so exciting.
Yes. Yeah, I was gonna ask about the progesterone as well.
I want to
awesome.
so badly, especially with the cycle differences. Something the uterine horns are actually different between the two of them too. The ovaries are different. There's a lot there with the hormones that I haven't even gotten levels yet. So we'll see.
Okay, well, we'll definitely talk more at SPM.
Yeah.
So,
Bye.
that's perfect. All right, well, thank you so much. Thank you so much.
Thank you.
All right. Well, let us move. So we're planning for 5 presentations today, but we don't have one of our presenters, so we have 4 presenters today. And so last but not least, we have Christy Macleod. Christy is a PhD student in clinical psychology at McMaster University, supervised by Benicio Frey and Dr. Benicio Frey and Dr. Nicholas Bach. Her research uses structural magnetic resonance imaging to examine sex-related differences in the microstructure of the cerebral cortex. Currently, her research is investigating sex-related differences in intracortical myelin in individuals experiencing major depressive disorder, as well as examining potential functional correlates in cognitive abilities and depressive symptoms. She is also conducting a longitudinal study to examine potential changes in intracortical myelin amount and distribution based on response to psychopharmacological treatment. So today, Christy will be giving a talk titled Examining Sex-Related Differences in Intracortical Myelin. Awesome. Well, thank you so much, Christy, and go right ahead.
Thank you for that. It's been really great to hear everyone else's exciting research, and I'm really thankful to be here and to be able to share mine. So as previously mentioned, I'm supervised by Dr. Benicio Frey and Dr. Nicholas Bach. Dr. Frey is a psychiatrist at St. Joseph's Healthcare Hamilton. He works in the mood disorders clinic as well as the women's health concerns clinic. And Dr. Nicholas Bach is a neuroimaging researcher from the psychology department at MAC. And so today I'm going to be presenting some of the past and ongoing research that I've been conducting while working in their labs for my PhD so far. And so to start, I'm going to give a brief overview of major depressive disorder, or MDD, which is a major focus of my presentation today. So major depressive disorder, or better known as clinical depression, is one of the most prevalent mental health disorders worldwide, and it causes severe impacts on the day-to-day life of many individuals. The cause of depression is still not fully understood, and there's quite limited efficacy of treatment. So its symptoms include low mood and decreased ability to experience pleasure, as well as changes in sleep, appetite, energy, and subjective cognitive abilities. Depression is a major concern for women's health due to the fact that women are twice as likely to be diagnosed with MDD when compared to men. So I think understanding this sex-related difference might be very important because it might give us insight to the etiology, risk factors, and symptomology of depression. And so my area, as previously mentioned, of research looks at a substance in the brain called myelin, which is a lipid-rich protein that coats the axons of neurons and allows for a quick and efficient signal propagation within the brain. So most myelin is found within the deeper part of the brain called the white matter, and that's been studied pretty extensively, but much less is known about the myelin found within the cerebral cortex, so these outer layers here. And this is what we call intracortical myelin, or ICM for short. ICM is hypothesized to play an important role in the synchronization and stabilization of functional brain networks. as well as several studies have shown that ICM may be involved in cognitive tasks as well. Importantly, recent studies have shown that there are deficits in ICM in the prefrontal cortex of those with depression and other mental health disorders. And studies in bipolar disorder have linked deficits in ICM to impaired cognitive performance. So looking at all of this, increasing evidence that ICM is involved in the healthy functioning of the brain, we conducted a study to explore if there were sex-related differences in ICM in healthy young adults. And so with advances in neuroimaging, we're now able to measure a proxy of ICM in vivo, which allows us to map the amount and distribution of ICM in a large data set, as can be seen in this group average map over here. And so we parcellated the cortex into 360 regions, and then we ran an analysis looking at the effects of sex and age on ICM. And so after correcting for the multiple comparisons, we found a significant effect in 320 out of the 360 regions of the cortex, all of which with men showing higher amounts of ICM compared to women. So in this figure here, you can see anything that's kind of in red, pink, or this light kind of white here are regions that are significant for men showing more ICM than women. These light gray areas here are where it's thresholded because it's not significant statistically. And so this sex-related difference in ICM has led us to a lot of questions about its potential relation to the sex-related differences that we see in depression. So namely, does having less ICM make women more vulnerable to the effects of MDD? Are the deficits in ICM related to cognitive impairment in MDD? And this led us to my ongoing research right now, a study that's looking at, for the first time in living patients, how ICM is disrupted in MDD, and if there's sex-related differences in both ICM and cognitive abilities. So our study is recruiting young to middle-aged adults that are currently depressed to study the brain composition in these participants. We're using the aforementioned neuroimaging technique as a surrogate marker for ICM in the brain. And we're also doing cognitive testing to look at abilities like processing speed, attention, memory, and other things such as that. And so we'll be comparing males and females with MDD to healthy matched controls. And then because research suggests that psychotropic medications can have a supportive effect on ICM and mental health disorders, we're also going to be collecting longitudinal data to explore if there's a pro-myelinating effect of antidepressants. So essentially, it's our goal to understand the mechanism of these sex-related differences in the neurobiology of depression. And this might allow for the potential for sex-specific treatments to improve outcomes for women with MDD. So thank you all very much for your attention, and I welcome any questions that anyone has.
Awesome, thank you so much. Awesome. Does anyone have any questions right off the bat? Oh, yes. Victoria. There you go.
Wonderful talk. It was very concise and brief, and you might have mentioned this, but I'm interested in, you had said that your ongoing research is going to collect data from young and middle-aged participants. Mm-hmm. And that got me thinking a little bit about if there are any age-related differences in ICM, and if you know if any of those potential changes with aging might interact with sex.
Yeah, so that's a great question. So there's been a lot of lifespan studies. I actually have a couple of slides I can show maybe that will be helpful for this. There have been a lot of lifespan studies that look at ICM across the lifespan, and there is actually quite a replicable effect of age. And so let me just show this slide quickly here, you can see. So there is this kind of protracted development of ICM that continues, that kind of continues into the third decade of life, which is surprising because most folks consider that the brain is fully developed by your early 20s. But we actually show that, we actually see that that's not the case, and that ICM continues to develop well into the middle age. And so that's something that we're interested in as a function of depression as well. So kind of understanding if there's kind of premorbid deficits, or if it's something that we're seeing after the first onset of a mood episode. And so we're interested in looking at that. And if we can see any changes based on people's response to antidepressant treatment.
Wonderful, thank you.
Oh, perfect. Any other questions that anyone has? One question that I was thinking of, definitely very interested in, are you going to be examining also specific symptoms that the patients have, and what symptoms might tie best to, or might be tied somewhat directly to ICM? I'm wondering what role it might play in, because since there's so many symptoms of depression and they kind of all form together, but some patients have certain symptoms, some patients do not have others. I'm wondering if you're going to be examining that.
Yeah, so that's an excellent point. MDD is, notoriously heterogeneous. It's a very complicated disorder. And so we are, looking at a lot of other things as well. So we're looking at kind of symptom profiles. We're taking blood for, for a couple other things we're looking at as well. So we will be looking at the symptom profile in association with some of these things. And, we're kind of hypothesizing that it might be related to neuropsych testing on cognitive abilities, but we also wonder if that might, have to do with subjective cognitive abilities that the patients are reporting, with their presentation.
You are going to have so much data. data. It's awesome. Love it. So much work. Okay, awesome. Thank you so much. Thank you. Any other questions before? And any other questions for any of our speakers today that have gone? Awesome. Some amazing talks today. I want to thank you for so much for your talks. Before people start to run away, I just want to quickly share, and then also I said before that I was going to share a charity for helping Indigenous people, a link that will guide you to some charities for helping Indigenous peoples and Indigenous women's health opportunities. So I will put that, actually, I will stop sharing right now. I will put that in the chat right here. And also, I'm also going to say, so we also have another session coming up at 2 p.m. today on MyTax, which I'll put another link in there for everyone. And now I'll share my screen just so you can see what it's all about. That we will have another presentation later on today at 2 p.m. PST. So feel free to still register for that session if you are interested in learning more about MyTax. Also, our next training presentation series, in case you're wondering, is aimed for September, and we already have a few speakers with tentative titles that are going to be speaking at in September's presentation series. So we're going to have talks on Parkinson's disease, talks on doperidone and low breast milk supply, as well as progestin, progesterone, acetate, and a rat model of surgical menopause will be coming up in September. So very excited for those talks. Super happy to have everyone here for these talks. Hopefully you keep coming to our trainee talks. I love to think of them as just a chance for all of us to just chat together about our research, and you all are doing such exciting things. I love hearing about all that research every time we share. Also, we always like to put a link out for donations for our research health cluster. We're applying for funds again, but just in case, thinking of donations as well. But for everyone that is here, thank you so much for coming. I wish we did this more often. We should have more events sooner, but thank you all, and thank you again for our presenters. Thank you so much. Awesome. Also, thank you, Stephanie.